Okay, to finish up the notes real quick, we're talking about how big molecules or polymers are formed by putting together monomers. And so in my picture, I have drawn 4 monomers, and they are all bonded together to form a polymer. How are those formed? Those are formed when we remove, so every time 2 monomers come together, we remove water. So if I have 4 monomers and they are bonded together to form a polymer, that means that we had 3 condensation reactions, 1, 2, 3 condensation reactions, and we removed 3 molecules of water, and therefore by removing the waters, we allowed ends of those molecules to be able to bond together and form that bond, creating a polymer. And so we remove water to build a polymer. Okay, so we're going to take water away, but we're going to get a bigger polymer. And sometimes that's a little twisted in your head. You think that you want to add something to make it bigger. Well, you're adding monomers together, but to get the monomers to stick together, you have to remove water, and then it allows them a place to hold on to each other. And so we remove water to build a bigger molecule or build a macromolecule or a polymer. That is known as condensation reaction. It's a reaction, we're changing the molecules and we're taking away water in order to create a macromolecule or a polymer, a larger molecule. So for the example, sucrose is a sugar, table sugar. In order to get sucrose, which is right here, it is 2 monomers, a glucose and a fructose, bonded together. Glucose and fructose are fine just by themselves. The only way you can get them to stick together is by removing a hydroxyl, an OH from one, and a hydrogen from the other. That molecule will come out as H2O, water, and then it will allow the glucose and the fructose to bond together in what we call a glycosidic linkage, or a bond between sugars. All right, so a plant is going to do this. For example, a potato plant. They go through photosynthesis, they create lots of glucose, and then they have to store that glucose in their potatoes. But they don't store it as glucose, they store it as starch. Starch is an example of a polymer of carbohydrates. A lot of monomers, a lot of glucose molecules bonded together through this condensation reaction in order to form starch. So starch is an example of a polymer. All right, what about when we eat that starch? We actually have to get energy from glucose, but starch is a big chain of glucose bonded together. So the first thing that you have to be able to do is we have to be able to break down those starch molecules by dividing, by breaking apart the glucose, by breaking apart the monomers that are joined together in a polymer. We can then get to a single glucose, which is what we need in order to get energy, like all living things. So how do we do that? Well, in order to go through the breaking down, so if we're going to break down, so I'm going to highlight this or outline right down here, it's where our focus is now. I'm going to outline that in red. Hydro is water. So when we talk about hydrolysis, hydro is water and lysis is to break. So I'm going to erase this real quick just so I have some room to draw. Okay. So now we're going to focus on breaking down a molecule. So if I have 1, 2, let's just say I have two, a glucose and a fructose. I have them bonded together in sucrose. I eat the table sugar. What my body needs to be able to do is break that apart. So I have to be able to break apart the monomers in the polymer. Remember, poly is many, mono is one. So I have to be able to break apart this bond right there. Well, how do I do that? Well, the best way to do it is to do the reverse of the condensation, is to have water. This is why you need water. If I have water and I add water, I use water, which, by the way, hydro is water. Lysis is to break. If I'm going to use water to break something apart, I would call that hydrolysis. Hydrolysis. Hydrolysis is the breaking down of polymers by breaking apart the monomers. If I add the water back in, then the OH is going to attach to one of the monomers. The hydrogen is going to cap the other. And what I'm going to do is I am going to break those monomers apart. Digestion starts with hydrolysis reactions, or adding water to break molecules apart. All right, so last thing that I want you to think about here is the idea that energy is involved here. And so energy, when we talk about cellular energy, you have to know that cellular energy is ATP. ATP is adenosine triphosphate. Right here, I have an ATP. It has 1, 2, 3 phosphate. That is a charged molecule. That molecule has immediate energy ready and available. When it uses that energy, what happens is it pops off 1 phosphate. And so if ATP is charged, then ADP, okay, this is 3. ADP is adenosine diphosphate, so that's just 2. This is uncharged. We're going to learn about how ADP gets charged into ATP. And one of those things that we're thinking about is if I'm going to charge ADP, then I need to add another piece. I need to add 1 phosphate to another phosphate group. And in order to do that, if I'm going to build a molecule, I'm going to actually take away water. It's going to be a condensation reaction. However, the hydrolysis of a phosphate group releases a lot of energy. And so how do we get this energy? Well, hydrolysis, I throw water at it, it breaks a phosphate off because hydrolysis is breaking apart. So the breaking down of sugar drives the formation of ATP. The breaking down of ATP by removing a phosphate is where we get cellular energy. ATP losing a phosphate is where we get the energy to sustain our life. What I need you to do now is go ahead and continue with the vocabulary that you guys have as an assignment on Canvas. You also are going to walk through an ed puzzle. That's also an assignment. There's 6 questions. Please make sure you go back and make sure that you get those correct. Some of it will be pretty much repeat from what we talked about. And then when you're done with that, you're done with the material. And then all I need you to do is complete the form, which will be used for your attendance for this class. Hope you guys had a great day. If you have any questions, please send me an email. Sorry I couldn't be there with you today. I will see you on Friday. Tomorrow, you are going to be working on some notes that I post onto Canvas. Hope you guys have a wonderful day. Welcome to Secret Biology, and I'm excited to work with you through second trimester. Have a great day, you guys.